All right, so for the next 4 days in class, we're going to be fitting species distribution models. More specifically, we'll be fitting correlative species distribution models. So these are ones in which we use a statistical model to relate observations of a species to the environmental variables that predict that species range. So the basic idea in this framework is that we've got some locations where we've gone out and we've surveyed for a species, and we found it to either be present or absent at each of those surveyed locations. Alongside those data, we have observations of various environmental variables at each of those locations. These environmental data could be collected at the same time as the presence-absence data, or maybe they're from satellites or from other surveys. Then we use a statistical model to model the probability that a species is present based on the environment. The range of environmental conditions for which we expect a species to be present, more often than not, are what we'll call the niche of the species, but we'll get into what that means later. So now with this predictive model in hand, this model which relates environmental conditions to probability of occurrence, we can use data from across the landscape to infer which areas are suitable habitat for the species. And with future projections of the environment, we can infer which areas will be suitable habitat in the future. There's a number of methods we could use to do this, but in this class, we'll be focusing on likelihood-based methods, which we're all fairly familiar with by now. So these include maximal likelihood models, as well as Bayesian models, which we haven't gone over. We're going to focus on maximal likelihood in this lecture. So as we know, for any likelihood-based method, we have to assume a statistical distribution of the data follow. For data containing either species occurrences or absences, the distribution that we'll always use is a Bernoulli distribution, which is a simplification of the binomial distribution, and we should be familiar with both of these distributions by now. If a random variable is distributed according to Bernoulli distribution with a parameter p, then it equals 1 with probability p and 0 with probability 1 minus p. So in this case, p would be our estimated probability of occurrence. 1 would be the outcome that the species does occur at a given site, and 0 would be the outcome that the species does not occur at a given site. The simplest possible species distribution model is one where we have some species. So I'm going to say that species is rainbow parrotfish here. And we've surveyed sites along gradient of environmental variables, or just one environmental variable, in this case, temperature. And we want to estimate the probability that this parrotfish occurs as a function of the temperature. So we can see here that it tends to occur in this fictional data set, so this isn't really rainbow parrotfish. We can see that it tends to occur for higher temperatures and tends not to occur for lower temperatures. So just like we did with linear regression, we can make the parameter p of the Bernoulli distribution a linear function of an intercept alpha and a slope beta times temperature, which is x here, right? The only issue with this is that this could produce probability of occurrence values that are greater than 1 or less than 0. So we'll add this transformation, which is the logistic transform, which will shrink probability values to within 0 or 1. It'll produce this really nice sigmoid curve for us, and it's intuitive. If we're already at high temperatures and the probability of occurrence is already close to 1, this species has here no thermal maxima. So increasing the temperature isn't going to increase the probability of occurrence much more. There are diminishing returns here. The inverse function of the logistic function is called the logit, which takes the probability and transforms it to log odds. So we can express this model as the log odds of occurrence being a linear function of temperature. In other words, this model is still linear, it's just linear on the log odds scale. Of course, we know that species do have thermal maxima, and therefore we generally expect a species' probability of occurrence to display a hump-shaped relationship with temperature. It occurs for some intermediate temperatures, and then becomes less common as we increase or decrease the temperature away from that optimum. We can adjust this basic Bernoulli model to accommodate this by changing this from a linear relationship to a quadratic one. so that we're fitting a parabola on the log odd scale, which will form this nice bell-shaped curve on the probability scale. However, this is still a linear model as far as the computer is concerned. If we, for example, take the square of the temperature before passing it to this model, then all the model sees is that it's fitting two independent slope coefficients, one for temperature and one for the square of temperature. So this is still a linear combination of terms, and therefore we still refer to this as a linear model. The name for models like this, where we have a response distribution, such as the Bernoulli, and where a transformation of the parameter or the parameters of the distribution depends linearly on some combination of predictors, is called a generalized linear model. The function that connects the parameters of the distribution to the predictors is called the link function, and in this case, it's the logit link. The nice thing about this type of model is that it's a simplification of the models that we fit when we were doing maximum likelihood estimation in the last section of this course. With those models, we fit all sorts of complex functions like population trajectories, stock recruitment curves, and functional responses. With generalized linear models, we're just fitting linear combinations of parameters. What this means is that we don't have to write our own custom functions to run generalized linear models, like we had to with the custom maximum likelihood models that we ran in the last section of this course. The functions to run generalized linear models are already built right into R. And there's a variety of things you can put in this model and still have the model be a linear combination of predictors. So these include not just linear and polynomial terms, but also other unrelated predictors, which can be continuous, like temperature or depth or precipitation, or they can be categorical, like, for example, habitat type or soil type. And we can get a lot crazier with what we consider a linear combination of predictors. And from models that are inherently linear under the hood, we can produce extremely non-linear looking model fits, which is what we'll be doing during the next couple of labs as we fit models which are known as generalized additive models, or GAMs. Consider this example data here, which we'll use, let's assume a normal likelihood function with a mean mu of our normally distributed y variable being a function of our x variable. The relationship between y and x is wiggly here, and we can try to fit this with a polynomial, but when we use a GAM, what we'll be doing is modeling the parameters in our response distribution as linear combinations of what we'll call smooth functions or smoothers. And because we're using these smooth functions as model terms, GAMs are able to fit really complex relationships in very flexible ways. But it seems like a bit of a stretch to say this is still a linear model, right? Well, the key here to keeping these apparently very nonlinear models linear under the hood is how these smoothers work. Each smoother is a linear combination or sum of k basis functions multiplied by some weight that is assigned to each basis function. And each basis function is just a polynomial of our predictor, so like x squared, x to the third, x plus x to the fourth, so on and so forth. And so these smooth functions are themselves linear combinations of terms, and what we call generalized additive models are really just generalized linear models. So this particular type of smooth function is called a thin plate regression spline, but there are other possible types that don't all work this way. Let's see how this works. Here we have fitted the GAM curve in red, and the basis functions plotted as dotted lines of various colors. By selecting appropriate weights for each of these polynomials, we can get a function that sums to the fitted red curve. So for example, look at this black cubic function here, which in this plot spans from about y equals -1 to about y equals 1. If we give this function a negative weight, so that we flip it across the y-axis, then it's already fairly close to the red function. And we can adjust the weights of the other polynomials in a similar fashion until they sum to this red curve. We've got 9 basis functions here, but we might not need to use all of them to get a good fit. So for those ones that we don't need, we can just set their weights to 0. The more basis functions that we have with non-zero weights, the more wiggly this function can be. And the fewer basis functions we have, the smoother this function can be. So in essence, GAMs work by first choosing these basis functions arbitrarily before even fitting the model, and then using a version of maximum likelihood estimation to fit the weights for each basis function. They automatically strike a good balance and obtain a curve that fits the data well, but isn't unnecessarily wiggly. We won't go into exactly how they do that, but the basic idea is to combine maximum likelihood estimation with some sort of penalty on complexity, so that models with lower complexity and a better fit to the data are preferred. So that's the basic idea behind GAMs, which is what we'll use in the next couple of labs. And I hope that you got out of that that even though they're very flexible and can fit complex functions, they're a very natural extension of what we've already learned about maximum likelihood. You could, if you wanted, build a function to make your own GAMs from the ground up, just as we did for other models in this class. So now I want to take us back to the ecological context of these models for a second, because I think it's important not to lose sight of that. Perhaps the biggest thing to understand about correlative species distribution models is that they are inherently limited, because we're using observations of where species currently are to infer the environmental conditions in which a species is capable of inhabiting. So we call the current range of environmental conditions that a species inhabits, it's a realized niche. And the range of environmental conditions that a species is potentially capable of inhabiting, its fundamental niche. And these are not necessarily the same thing. So even if the climate today, was the same as the year 2100's climate, then there's a good chance that many species would still have a different range in 2100 than they do now, because the realized niche now does not match their fundamental niche. There are areas that they could be that they are not yet occupying. So what can cause these two things to differ? We know that species range is at a minimum a function of environmental factors such as temperature, habitat type. For example, some fish inhabit muddy bottom areas and others are only observed in rocky reef ecosystems, as well as primary productivity, intra-annual variation in the number of daylight hours across latitude, and altitude, or in marine systems depth. But species ranges can also be restricted or expanded by other factors. So for example, the range of a species might be restricted by the presence of certain predators or competitors in habitat outside of their range. So individuals of that species may never be able to colonize new habitat depending on the species in that habitat, even if the species is fundamentally able to exist there, to survive and reproduce there. Mutualisms can also alter or expand species ranges. I can guarantee, for example, that any species of coral would have a very different range if it weren't for the dinoflagellate symbionts that provide something like 97% of the energy that they get from sunlight. And plenty of research shows that plants with fungal associations have very different habitable ranges than those without. Further, the phylogeographic history, or the evolutionary history of the species and its related taxa across space, might play a big role. As a fairly extreme example, species of reef fish which have evolved in the Indian Ocean may well be able to survive on Caribbean reefs, but might never have made it there anyway. So people frequently consider species ranges as reflecting only current conditions, but historical processes can be very important in shaping ranges. A related issue is dispersal limitation and physical barriers to dispersal. If you're a fish living on a reef along the coastline, even if there's habitat not too far away that's viable for you to inhabit, you and your offspring may never see that habitat because your offspring are larvae that drift passively in the water column and always get carried somewhere else by currents. Your species might experience a range expansion in the off event that a few larvae do make it up to that habitat and grow to adulthood. But that sort of chance event may occur every 100 years, right? So genetic data from a few fishes which range across the Indo-Pacific to the Eastern Pacific suggests that these sorts of chance events are actually really important sometimes. And when we don't account for important processes in the model, we can end up with species distribution models that are very poor at characterizing the geographic range of our species of interest. Here's an example from a paper published last year, which was itself criticizing the use of SDMs in another recently published paper. The authors for the original paper, built tons of different shark species SDMs. And the result was that for a number of the species, their estimated current ranges were completely out of line with the known biogeography and ecology of those species. It's really hard to just automate this process. You need to actually have some ecological knowledge about the species you're modeling, and you have to apply that knowledge to the model building process. So building a bad distribution model is really easy, which is why basic SDMs have been fitted for pretty much every species you can think of. This website, Aquamaps, for example, has range projections for tens of thousands of fish, marine mammals, and invertebrates. These can be really useful, but you shouldn't trust any of these range projections too much. So pretty much the most important thing you can bring to the table when you're building an SDM is ecological knowledge, because otherwise you're probably just going to be repeating the stuff that's been done already. To add a little more caution here, it's important to examine the fitted relationships between your environmental variables and the probability of occurrence for your species. You want to make sure that the data you're using are actually sufficient to capture the range of environmental conditions that define the niche for that species. So for example, in the top right here, I've surveyed for the species at a range of sites between 20 and 32 degrees Celsius. And the bottom right, I've surveyed a much narrower range of temperatures, but I've still captured the thermal optima and some suboptimal conditions. In the bottom left, I've surveyed only the lower range of environmental of temperatures. I've captured sites that are too cold and sites that are just right, but I haven't captured anything that's too warm. So when I fit a Bernoulli model with a quadratic term to the data on the right, I get pretty similar curves that seem sensible. But when I fit this same model to the data on the left, the model tells me that this species is only more and more likely to occur as we crank up the temperature. So you can imagine that the models on the right and the model on the left will produce very different range maps, both now and in the future. Failing to account for important processes that structure the range can also cause statistical issues, the most common of which is called spatial autocorrelation. Spatial autocorrelation generally is when there's a clear pattern in the data across space, so that data which are close to each other have similar values. So let's take this map of population density in the US, where counties with large populations tend to be nearby each other, or this map of elevation, where pixels of high elevation tend to be surrounded by other pixels with high elevation. So this isn't a problem at all. We expect that when we look at the species distribution, there will be areas where they're generally more common. The issue is when we fit our model and we've tossed in the predictors, like for example, temperature and depth, that we think explain where our species is. But when we plot the difference between our observed data and our model predictions, also known as our residuals, there's still a spatial pattern. So what this tells us is that the data are not independent across space, even after we've adjusted for the model. And we know that as with any method which assumes a statistical distribution, we expect those data to be independently distributed according to that distribution. Spatial autocorrelation isn't necessarily a problem for prediction, but it's definitely a big problem whenever we want to accurately estimate uncertainty. That means that whenever we want to compute hypothesis tests, confidence intervals, and the like. Now, in many cases, adjusting for spatial autocorrelation is pretty difficult. But the nice thing about GAMs is that we can do it very easily by just including two-dimensional smoothers across latitude and longitude. So these work the exact same way as the smoothers that we've already gone over, even if they seem conceptually a little more complex. And we'll get some practice with these while we work through our first SDM lab. So I'm going to wrap up this lecture, but I do want to say that there is a lot more to the modeling process, even when we're specifically talking about GANs. Here's a general flow chart for fitting SDMs of whatever type. You can see that there is a lot we haven't covered here. So where do we get the data from? How do we choose among models of different types or models of different variables in them? How do we decide whether or not the model we fit does a good job of explaining the known range of the species? These are all questions that we'll touch on in lab, but we'll just be scratching the surface. And of course, GLMs and GANs are not the only types of species distribution models. Any method that you apply to modeling a species distribution is a species distribution model. I've listed some common model types here just to make it clear that GLMs and GANs fit into one category. These models work in different ways and expect different types of data. So for GLMs and GANs, we're usually working with presence and absence data, but some methods only need to know where we have observed a species, not where we haven't. So that's presence only data. And some methods work by characterizing the conditions that we have observed a species in and comparing them to the background set of environmental conditions that are available, which we call presence background data. So that's opposed to characterizing both the environmental conditions where they're observed and the environmental conditions where they haven't been observed, which is presence absence. So choosing a model will often come down to either the data you've got or the purpose of the model. Maybe you just want a model with the best possible predictive accuracy, or maybe you want a model that can accurately characterize the environmental niche of the species, or maybe you've got some other goal entirely. And I'm not going to try to make explicit recommendations for different model types, but just know that frequently the model selection procedure comes down to model purpose. And on that note, see you in class.